Hello to everyone. Good afternoon. Thank you very much for inviting us to this event. We are Anna and this is Chantal, and we want to talk about Vespa velutina on the colonies of honeybees, and also we want to talk about the description of the nest. To begin with, we can place the arrival of the velutina in Galicia, located in the northwest of Spain, in 2012. Since then, it has spread throughout the community, especially in coastal areas. In Galicia, more than 28,000 nests have been geolocated in 2021, and more than 150,000 nests were reported to local government authorities from 2012 to the present. Vespa velutina is a species that exhibits social behavior with behavioral flexibility, and this may contribute to its introduction. Along with the above, the invasive success of this species is also related to the ability of a single queen to form a large colony and produce many reproductive females that later give it a high dispersal capacity. In addition, the abundance of Apis mellifera, of honeybees, throughout Europe favor the multiplication of this species, since it is probably its main prey, which still does not have a clear defensive behavior against this hornet. In this sense, one of the most affected sectors is the beekeeping sector. Velutina disrupts the activity of the colony and causes, among the other effects, a drop in the production and storage of reserves. Okay, so where are the reporting nests located? Our research group have analyzed the data from all these nests registered by the surveillance and control program of Velutina was in Galicia. And with the result, we have a work, a paper entitled Embryo Relocation and Secondary Nest of the Invasive Species Vespa velutina in Galicia, that I'm going to explain next. So nowadays, this hornet is present throughout the Galician territory, but 59% of the total nest were from altitude rated low below 200 meters, and 16% at altitude above 400 meters. This implies that the coastal areas were the busiest. Okay, regarding the nest place, about 61% were found in vegetation. The most common were deciduous trees like alder, poplar, walnut, chestnut, oak, willows, and fruit trees. Within the perennial trees, there were usually eucalyptus globulus mainly, and then they use conifers too, such as Pinus, Cedrus, Picea, and Cupressus. Furthermore, nests were detected in Laurus nobilis, Ligustrum, Olea europaea, and Citrus, among others. The rest, 36%, were found in buildings, and 3% in the ground, in soil. Most of the nests were found in buildings, were discovered in the spring, coinciding with the development of the embryo nest. This means places like a roof area, unused attics, walls, waste containers, chimneys, windows, balconies, or even beehive. Okay, so how we can differentiate or describe this nest? We can differentiate two types of nest that differ according to the stage of development in which they are found. Um, Thus, we have the embryo nest of an early stage and the secondary nest of an advanced stage of development. And it is common for this species to relocate its nest elsewhere. In the sense, we can speak or we can talk about relocated nest. Okay, the structure of the embryo nest starts with a single point of union that joins it to the surface, called the petiole. We can see in this video, around this point, the queen makes the first comb of hexagonal cells. This structure is quickly surrounded by a soft, loose, and straw-like cover forming an envelope. Then new layers are added, turning around each other in a spiral. The number of envelope layers increase as the nest is constructed. We can differentiate at the bottom a hole of 1.5 centimeters that serve as an entrance. During this period, before the first worker or males emerge, the queen builds the nest along having a mean cells building rate of around 1 cells per day. When the first individuals emerge, the cells building rate increase until 3 cells per day. We can highlight that we have found males in this kind of nest, in nests a little larger than this. On the other hand, the perimeter of the embryo nest varied or ranged between 8 and 33 centimeters, and then the nest height ranged from 3 to 10 centimeters. Internally, the nest can have one or two combs supported by small petioles. For the first combs, the area can vary between 2 and 66 square centimeters, and for the second comb, the size was between 2 and 24 square centimeters. Secondary nest had a consistent outer cover composed of compacted layers. Frequently, this cover is more than 5 centimeters wide. The color varies from light straw to dark green. The internal structure is formed by the combs joined with petioles. Nests have one main big lateral entrance and a small secondary entrance situated from the middle to the bottom. Studied secondary nests were from different periods of the biological cycle, thus they have different development stages. The smaller one had a perimeter of 26 centimeters and a height of 8 centimeters, while the bigger one had 162 centimeters of perimeter and 120 centimeters of height. Regarding the number of combs, this varied from only 2 combs to 11 combs, with a mean number of 5 combs. Okay, and the smaller nest had only 33 cells. 20 1,000 cells were calculated for the largest nest. This gives an idea of the number of individuals that can leave a nest. The mean number of cells was 4,800 cells. The nests grow larger as the season progresses, thus the number of combs and therefore the number of cells increase continuously. Larger nests collected in autumn and winter containing thousands of cells. And then early spring nest and the nests at altitude above 400 meters had the smaller cell area, while autumn and winter nests showed higher cell area values. The area of the cells varied from a mean of 0.39 square centimeters for spring nests to a mean value of 0.56 square centimeters for cells of winter nest. We have to say that the cells area determines the size of the hornets. So hornets from a smaller nest are smaller than hornets emerging from secondary nest. As for the shape of the combs, it follows the patterns of the nest, adapting the size and shape to the place where it is located, as well as the presence of elements such as tree branches or other structure used to support the nest. On the right, we can see a picture of a nest. The photo is upside down since the nest will be in that position in the environment. Normally, from the first comb, that often has a value over 250 square centimeters, to fourth comb, the area increased progressively. 5 has the highest variability inside, and from comb 6 to the last comb, depending of the net side, the area decrease. The smaller nests are spherical in shape, but as they get larger, they become more oval. Nests grow in winds and haze during the whole growing season. Okay, so regarding to the relocation nest, these nests are characterized because they were found in early summer and the combs were empty of brood or meconium. The number of combs in these nests range from 1 to 3. However, one of them presented the external structure, but without any combs inside. So we have found in the interior of these kinds of nests around 50 hornet female, um, and we found also eggs, but no meconium, larvae, or pupa were observed in the combs. This observation can suggest that the odor found inside of the nest came from a different nest, a different part. Okay, so we could use meconium to estimate the population of the nest. First of all, we have to say that meconium is the fecal matter that accumulates in the gut during the larva development, and it indicates that hornet were raised in the combs. More than one layer of meconium was observed in cells, both in the embryo and secondary nest. The weight of meconium in the embryo nest was around 40 milligrams, like a mean value, that is lower than meconium in secondary nest, that was 60 milligrams. In the middle of the first combs of secondary nest, the cells are used up to 3 times, and in the embryo nest up to 2 times. Finally, to estimate nest population, the total number of eggs, larvae, and the calculated emerged hornet from the meconium were considered. With this, we can see that the laying capacity of the queen is lowest at the beginning of the life cycle and also at the end of the life cycle in the winter. The maximum population is reached in autumn and winter. In this table, you can see an estimated value of 30,000 hornet in a autumn nest. That is why we can explain the great pressure they have on honeybee hive.
Hello, I'm Anna, and I'm going to talk about pressure on apiaries in Galicia. Also, I'm going to talk about the pressure that we analyze in different apiaries in all the territory, and which systems we use for measuring this pressure. And also, I'm gonna talk in the last slides about the most important methods for us to avoid the colony losses. So the first that Chantal told you is that in Galicia, Vespa velutina is present throughout the whole territory. This map is a map of the Xunta de Galicia, is a map of nest warning in 2022. The colors, the first colors are refer to the lowest number of notified nests, and as you can see in the next picture and the next map, these colors matches with the places with the higher altitudes in our territory. So that means that the number of nests of, and therefore the pressure on apiaries will depend on the altitude and climatic characteristics of the area. Okay, in this map, I will show you the apiaries that we monitor. We monitor more than two, but we analyze only two until now. So I'm gonna talk about two of them. We analyze two apiaries in the year 2021 and the last year in 2022. One was in Sergude. It was in a altitude of 260 meters, this apiary, and the second one was in the coastal margin of Galicia in Ribeira, and the altitude was 5 meters. Each apiary had different weather conditions and hornet pressure each year. So now what I am going to talk about is the systems that we use for measuring the pressure and also the weather on the apiaries. We place a camera in front of the two colonies. Also, we did the same in the other apiary, in both of them. And also we put an external system that measure temperature and relative humidity, and it was placed around the apiary next to a tree or something like that. Both of them measure the parameters every hour, and the camera runs from 7 a.m. to 10 p.m. And so the night we don't have any picture of the hive entrance. To understand better what's the behavior of the hornet and also the behavior of the colonies, we analyze the managed methods that the two beekeepers use in these 2 years. In Ribeira, no methods against hornets were used in the 2021, but in Sergude, electric ARPs and queen excluder were used. As you can see here, the electric ARP was next to the two colonies, and here the queen excluder was were used when the pressure was higher in September, and was placed in the hive entrance to avoid that the hornets enter to the colony. Here we analyze the pressure in both of the apiaries. In green one is Ribeira, that was the apiary in the coastal margin, and also the yellow one that is in Sergude. We count the number of hornets that the camera or the picture show us in each hour and each day. So what we saw is that the pressure started before in Sergude, but a higher number of hornets started in Ribeira. Then we saw that the highest pressure in Ribeira was in July, while in Sergude was really low, the number of hornets this month. And what happened in September is that in Ribeira, both of colonies died. The beekeeper and we didn't use any kind of control methods against hornets. But we continued the study in Sergude, and we saw that the highest pressure in Sergude was in October, when the temperature and humidity was lower and the humidity was higher. And the last hornet seen in 2021 in the pictures in Sergude was the 25th of December. That means that the pressure on colonies extend until December if the temperature and humidity are nice for them. The two colonies that die in Ribeira, we replaced them and put two alive colonies for the second year of the study. The management methods that we used in Ribeira this time, it was the queen excluder between the brood chamber and the bottom, as you see in the picture. And in Sergude this time we use, and the beekeeper used Trojans, also electric ARPs and the entrance reducer. And what we see this year in Sergude is that the beekeeper put earlier the reducer that provoked that all the drones die in the colony, and we have this problem in the hive. All the drones were in the colony and the workers could not get out the drones, and they had a lot of hot in the colony and provoke dies. Also the electric arc was sound, and it was really disturbed for the honeybees. All the drones on the floor. In this year, in 2022, in both apiaries, in all the colonies, they were fed if it was necessary. So what happened in July in Sergude was that the beekeeper made a starter colony, and that provoked, as you can see in the right in the picture, that this small colony was weak and caused that a large number of hornets go to the entrance of the colony and enter in the colony and cause a lot of damage on it. And in this graph, you can see what happened in the in Sergute. That remember that is the yellow one, the yellow line. That in July, in the middle of July, a really high pressure appeared in the apiary. That it was this starter colony in this one. So the pressure in both apiaries this year started later than in the previous year. Also in the beginning of July. Then one of the colony was replaced for the small colony that you see in the picture of the slide before. This provoked that the highest pressure in Sergude was in July. This colony, of course, died, and also the colony next to it. Then the beekeeper decided to use Trojans as control method. In the other apiary in Ribeira, the highest pressure was in September this year. But despite the use of Trojans, the hornet pressure was higher in Sergude than in Ribeira. So once the the beekeeper replaced the colonies, we have the same pressure as the last year. So what happened in Sergude is that the two colonies died in October this year again, and the two colonies died in October in Ribeira. So now I'm gonna show you some videos about the pressure of Vespa velutina on apiaries in Galicia and how it starts, how it continues, and then how it starts with the beginning of the colonies. So here you can see the pressure, how it starts. Here I'm gonna show you how the pressure continues. This colony, you see it, had a lot of population. So the hornets were hatching the honeybees that came from outside of the colony. Now I'm gonna show you how the pressure is really high, but the colony is still alive. And here is a colony that died because of the pressure of Vespa velutina. And now what we want to show you is that the pressure is different throughout the day. For example, in Sergude, in both years, that is the left graph, you see that in the midday, less hornets are in the apiary, and the highest pressure is at 9 p.m. and also at 11 a.m. For the contrary side, or for in a different way, in Ribeira, the highest pressure is in the midday. Also, the temperature is lower. This happened in July. In August, it happens the same in Sergude, so it has less pressure in the midday, and also the temperature was higher at this time. But in Ribeira, the highest pressure was in August, and it was in the midday. And in September, the pressure was higher in Sergude in the midday. Also, the temperature was lower. And in Ribeira, it has a irregular graph of the pressure of Vespa velutina. What I want to say with all of this is that the hornet's pressure is different throughout the day and also throughout the year, and it depends on the climatic conditions. And now I'm gonna talk about the another part of our work, that is the precision beekeeping, that we use for a better understanding of the colony and what happened with the colony with the pressure of the hornet. We use uh, scales that measure the weight of the colony, and also we use an internal sensor or internal system that measure the temperature and the relative humidity inside of the colony. This measure the system measure the parameters each 10 minutes and send to a computer host that we can see in our house. What we saw in our study is that the thermoregulatory capacity of the colonies is gonna show us, or is gonna tell us some information about the strength of the colony. It was shown, like in the pictures that I showed you before, that the weakest colony had more hornets pressure than the strongest one during all the period in this paper or this article, and also in what we saw in the apiary. So maintain a high colony strength for us and for the beekeeper, it should be the most important managed method to avoid the colony loss. Because it is known that the hornets choose weakened colonies to forage the food because it's easier for them. So what a beekeeper can do for maintain the high colony strength is, for example, to use entrance reducer, as you can see here, or for example, a queen excluder like this on the entrance, but also between the brood chamber and the bottom. With these two methods, the beekeeper should be careful, because if you use the entry reducer or the queen excluder in the time when the temperature is high outside, or the brood has a lot of drones, it could happen that the workers cannot throw the drones outside the colony, and as you see in the video before, they are gonna have a lot of temperature inside of the colony. And also, if you forget that you have an queen excluder between the brood chamber and the bottom, or in the entrance, you are not gonna allow the colony to swarm. So, as you can see here, the swarm, and also all the drones that die, and also workers on the colonies because of that. Also, what the beekeeper can use, mussels like this. As you see, the hornet goes here and they cannot escape and they die in the mussel. Also, the beekeeper can use electric arcs. Be careful with that because it has continuous electricity and it's dangerous to use it, but it's a good method also. But for us, the most important and the cheapest one is to go to the apiary and check food and population stocks very often, and if it's necessary, feed the colonies. But if not, not protect the colonies in general. Like this, they need nectar, but also they can need protein. And if you have time and you have a really, really, really good eyes and vision, you can look for the nest around the apiary, but it's really difficult to find them in when the leaves are on the trees. So with this, we finish our presentation. Only we want to thank you again for listening and also for inviting us to this event. And that's it. Thank you.